warm welcome to all participants joining us from across India and around the globe. We are the students of People University are proud to present the special webinar designed entirely by the students and for the students. On the behalf of our organizing team at the Center for Scientific Research and Development at People's University, Bhopal, India, I warmly welcome you to the fourth session of the BioConnect webinar series. I am Nityansh Patel. I am proud to tell you that I am the student of BTech Biotechnology and today's your MC. It's truly a great honor to host this insightful event. Today's webinar focus is on a provoking theme, biotechnology and synthetic biology in the biosecurity era, opportunities, risks, and safeguards. As science advances, so do our responsibilities. This session explored how biotechnology and synthetic biology can be a powerful tool in ensuring a more safer, more relevant world, also navigating the ethical and the biosecurity challenges they present. We are deeply honored to welcome the guests and the leaders joining us today. A very special welcome to our Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, Dr. Leyma P.T. Haro, our speaker and our expert in biosafety and biosecurity, Professor Dr. Subhash J. Bore, head of the Center for Scientific Research and Development at People's University, and our mentor, Ms. Vanshika Trivedi, the chairperson of the organizing committee of this program. To formally inaugurate today's session, it's my honor to invite our Honorable Vice Chancellor, Professor Dr. Harish Rao, sir, to deliver the opening remark. Sir, over to you.
Thank you, Nityansh. A very good evening to all. It is my great pleasure to extend a warm welcome to each one of you to this special session of the BioConnect web series, an outstanding initiative led by our students for the students under the aegis of Center of Scientific Research and Development at People's University, Bhopal. At the outset, I would like to sincerely congratulate our brilliant BTech Biotechnology students for organizing this intellectually enriching event. Your proactive spirit, curiosity, and commitment to academic excellence are deeply inspiring, and today's program is a testimony to that. I would like to take a moment to commend Professor Subhash J. Bore, Head of Center of Scientific Research and Development and Student Advisor, for catalyzing this initiative, for his guidance and the leadership in bringing this idea to life. The theme of this webinar, Biotechnology and Synthetic Biology in the Biosecurity Era, Opportunities, Risk and Safeguard, is both timely and significant. We are living in an age where technological advancement in the life science is moving at an unprecedented pace. Biotechnology and synthetic biology are revolutionizing sectors ranging from healthcare to agriculture. However, with great power comes great responsibility. The integration of biosecurity into scientific innovation is not just necessary, it is essential. We are honored and delighted to have with us today Dr. Leyma P.T. Haro, a globally respected expert in biosafety and laboratory operations. Dr. D. Haro brings a wealth of international experience and insights, and we are eager to learn from her about the global trends, ethical considerations, and regulatory framework that govern this vital intersection of science and security. This session is more than a talk. It is an opportunity to ignite critical thinking, responsible innovation, and global dialogue. I urge our young researchers and future scientists to engage actively, ask bold questions, and explore how you can contribute to building a safer and more secure world. Before I conclude, let me once again commend the efforts of the organizing team and the CSRD leadership for making this event possible. Such student-led initiatives align perfectly with our vision of empowering learners leaders and fostering a research-driven academic environment at People's University. Let us continue to support and celebrate student innovation and scholarship. Thank you, and I wish you all an enlightening and a deep, immersive learning and impactful session ahead. Now I will hand it over to our MC, Mr. Nityansh Patel. Over to you, Nityansh.
Thank you, sir, for your motivating words. Your continued support to the students' academic initiatives is deeply appreciated. Now, I invite the chairperson of the organizing committee, Ms. Vanshika Trivedi, to say a few words. Vanshika, over to you.
Thank you, Mr. Nityansh. Hello, everyone. Our guest speaker, Dr. Leyma PG Haro, and Honorable Vice Chancellor of People's University, Professor Dr. Harish Rao, our mentor Professor Subhash Bore, respected dignitaries, esteemed guests, respected faculty members, and dear fellow students. My name is Vanshika Trivedi, and I'm proud to tell you that I am a BTech Biotechnology student at People's University. It is a true honor to serve as the chairperson of the organizing committee of this program. On behalf of the organizing committee, I warmly welcome all of you to this important webinar titled Biotechnology and Synthetic Biology in Biosecurity Era: Opportunity, Risk, and Safeguards. Today we live in a world where science and technology are growing very fast. Biotechnology and synthetic biology are helping us to create new medicines, better crops, and even solutions to protect our environment. These fields brings us great opportunity to improve our lives and solve global problems. However, along with these benefits, there are also risks. For example, powerful technology could be misused to create harmful biological agents, or mistaken experiment could lead to unexpected danger. That is why biosecurity, protecting people and nature from such risk, is very important. In this webinar, you will learn about both sides, the exciting possibilities and the serious responsibilities that comes with them. You will learn how scientists, policymakers, science can collaborate to ensure that these technologies are used safely and ethically. To my fellow students, I want to say you are the future of science. Be careful, be responsible, and always keep in mind the impact your work can have on society and nature. Use your knowledge not just for innovation but also to keep the world safe. I am confident that today's session will inspire us all to think deeply about how we can strike a balance between progress and precaution. With these words, I thank you once again for joining us. Let us have an engaging and thoughtful session. Now I hand over to Mr. Nityansh. Thank you.
Thank you so much, Ms. Vanshika, for your encouraging words and welcoming remark. Fellow students and esteemed participants, it is now time to begin the core session. To guide us to this topic and our keynote, I hand over the session to our moderator, Ms. Alisha Aslam. Alisha, over to you.
Thank you, Mr. Nityansh. Hello, everyone. A warm welcome to all those joining us from India and other countries. I am Alisha Aslam, and I'm proud to tell you that I am a BTech Biotechnology student at People's University. It is my honor to serve as a moderator for today's session, hosted under the BioConnect webinar series by the Center for Scientific Research and Development, CSRD, People's University, Bhopal, India. Today's session addresses a subject of great relevance in our rapidly advancing scientific world: biotechnology and synthetic biology in the biosecurity era: opportunities, risks, and safeguards. As biotechnology and synthetic biology continue to transform medicine, agriculture, and environmental science, we are also confronted with a new era of challenges, those concerning biosecurity, dual-use research, and the ethical governance of powerful biological tools. Navigating this landscape demands not only scientific expertise, but also global awareness and responsible action. To guide us through this critical discussion, we are privileged to welcome Dr. Leyma P.D. Haro, a distinguished expert in biosafety and biosecurity. Before we start, let me introduce Dr. Leyma briefly. Dr. Leyma received her BS in biochemistry from California State University, Los Angeles, and holds a PhD in biomedical sciences from the University of New Mexico. She is a registered biosafety professional, RBP, certified by APSA International, USA. Her postdoctoral research includes fellowships at Los Alamos National Laboratory, followed by significant work at Sandia National Laboratory, where she was part of the globally recognized Global Chemical and Biological Security team. Currently, Dr. Leyma serves as a senior scientist at Merrick and Company in Colorado, USA. With over 15 years of post-PhD experience, she has contributed significantly to enhancing biosafety and biosecurity practices worldwide. Her background in advanced biomedical research allows her to understand the complex safety challenges that laboratories face today, particularly in an era of rapidly evolving life sciences. Dr. Lema's work is driven by a deep commitment to building a culture of safety and responsibility, supporting laboratories and organizations in pursuing scientific innovation without compromising ethical and security standards. It is truly an honor to have her with us today. With this brief introduction, it is my great pleasure to invite Dr. Leyma to deliver her talk. Dr. Leyma, over to you.
Thank you, everyone. Can you hear me okay? Thank you, everyone, for inviting me to participate. And it's an honor to be here today. I am very excited about this topic. I think it's wonderful that you've created this webinar series with the initiative of the students and by the students, because you are the future of this field. And you are already taking leadership. So I have a very exciting topic to discuss today with you. I hope you will enjoy it as much as I do. Let me find my presentation, or I will share my entire screen. Can you see my presentation?
Yes, we can see.
Okay.
Mm-hmm.
Perfect. So as I present, please feel free to interrupt me, ask questions as we go along. Do not feel that you have to wait until the end to ask questions if something strikes you in the middle. So today we'll be talking about biotechnology and synthetic biology and sort of the risks, the opportunities, and the safeguards that we need to be thinking about as we move forward into the future, which is moving at exponential speed. Here's an outline of what we're going to talk about today. First, I'll give you a brief introduction to synthetic biology and biotechnology, although I think this will be very brief for this group. Then I will make a case for why we need to be thinking about biosecurity in this context. And then we'll talk about some new material and new ideas that I've produced recently. We'll talk about artificial intelligence, the role of humanoid robots in research, and lab automation. It sounds a little bit sci-fi and futuristic, but let me tell you, the future is here. Since this is an academic webinar, I decided to include some learning objectives today for you. So I hope that at the end of this presentation, all of you will be able to at least explain at a basic level what is synthetic biology and biotechnology, and that you will be able to identify at least some basic biosecurity risks that are associated with this type of research. And I hope that I will provide you some insights to help you anticipate future development and the potential risks and benefits of these future technologies that have not emerged yet. So biotechnology uses living organisms, biological systems, or their components to develop products and technologies that improve human life in the environment. It is a multidisciplinary field. It spans across medicine, agriculture, industrial processes. It can combine biological tools with engineering, chemical tools, and computer science, as I'm sure you already know. Synthetic biology, I view it as a tool within the biotechnology toolbox. Again, it's a multidisciplinary field that combines biology, bioengineering, genetic engineering, computer science, information technology. The aim of synthetic biology is to design, construct, or modify biological systems for improved functionality that will help address global challenges. For example, global health vaccine development, maybe eliminate or alleviate climate change issues, food insecurity, energy, among many, many other applications. So what are some examples of synthetic biology or biotechnology that already exist? One of my favorites, and the classic one, is the invention of insulin. So in the beginning of, well, not in the beginning, but let's say over 100 years ago, if somebody developed diabetes, chances were that they had a certain death after a short amount of time from disease because there was no cure or treatment. Eventually, some scientists figured out how to purify insulin from pigs' livers. And so purified pig liver insulin was delivered to some patients. This was extremely expensive and, of course, only available to certain people that lived within the reach of where this technology existed and that were able to afford it. So it was, of course, well beyond the reach of the reach of poor people or people that lived in more rural areas or less developed parts of the world at the time. But then came biotechnology and synthetic biology, where the human insulin gene was introduced in bacteria. And now most people who have diabetes inject themselves with a synthetically produced insulin that's produced in a lab with bioreactors. It's in bacteria, it's purified, it's highly consistent, it's highly reliable, it's highly safe. It's a fraction of the cost of what it used to cost, and it saved millions and millions of lives worldwide, both for type 1 and type 2 diabetes patients. A second example is the redesign of existing biological systems. For example, this may be a little bit controversial, but you can genetically modify or engineer corn with a certain gene that can produce a toxin to repel certain pests, so that you don't have to spray your crops with pesticides. This toxin would only be toxic to the pests, but not to the humans or the farm animals that might consume this corn. So then you can have higher yield cornfields without the need for chemical pesticide or with a reduced need. Another example is the efforts that have led to trying to produce biofuels. In which a system you can use yeast, where you genetically engineer the yeast with an ethanol gene. You research it, then you prototype it with the yeast, and then you try to see if you can purify ethanol. You can check the quality control. If it works, it could be applied for commercial uses, and if not, you can redo the entire optimization cycle over and over again. This technology is in development. In my opinion, it hasn't yielded as good results as we all hoped in the beginning, but it is one of the uses. One of my more recent favorites is mRNA vaccines and nanotechnology. As we all know, through the COVID-19 pandemic, mRNA vaccines were already in development, but were really catapulted into the forefront. And ultimately, the major vaccines that were delivered worldwide were based on this mRNA technology, in which you put an mRNA inside a nanoliposome that then gets injected. And then the mRNA can be delivered for a vaccine, although the example I'm using here is a nanoliposome that could carry an mRNA selective to attack cancer cells or to modify immune cells to then attack your cancer cell. There are several such vaccines for cancer treatments that are in development right now in the UK and the United States for approval for use in humans. In my opinion, this type of technology will really result in developing cures for many diseases that have been considered uncurable up until now. Other examples of this are a treatment for sickle cell anemia and beta thalassemia, two conditions that were considered incurable up until very recently. And now they're treatable, potentially even cured if we let enough time pass by to see whether the patients are really cured or whether they will need to be re-injected with a treatment periodically. But either way, the lives of these patients have been significantly improved by these developments. This is a little bit visionary. It hasn't happened yet, but there's certainly many laboratories across the globe trying to work on this issue, which is synthetic tissues and organs, which would eliminate the need for live organ transplants or waiting for someone to pass away and have their organs harvested. So in this example, I'm using the example of a liver, which could be 3D printed using maybe even your own genetic makeup, your own genetic signatures, maybe your own stem cells to regenerate in the future in a 3D bioprinter, a brand new liver that would then be delivered back to you as the patient. So why biosecurity? Because all of this technology, which is very wonderful in my opinion, and can help to solve many world problems, also carries risks that we need to address. It doesn't mean that we should stop doing the research. It just means that we have to be good stewards of the knowledge that we are acquiring and of the technology that we are developing. So there's always a concern for bioweapons, which is that when fallen in the wrong hands or in the hands of someone with nefarious intents, they could theoretically use these technologies that we're developing for the benefit of humanity for harmful purposes. So they could create biological agents that could be weaponized or create new toxins or enhance already existing toxins that could then be used for harm, that are designed for the intention of creating harm. There's also the issue of bioterrorism, where these technologies could be potentially used in bioterrorism activities, for instance, releasing an engineered organism into an environment or trying to disrupt food supply, water supply, other things. It doesn't necessarily mean trying to create an organism that would make humans sick, but they could disrupt also the bioeconomy, agriculture, water, air, many, many things that could also have significant impact on a society, on a population. There's the unintended consequences, with one of the introductory students alluded to in her introductory remarks, which is that there could be the accidental release, meaning we're not intending to release these synthetic organisms, but they could escape the lab. And then they could have unintended consequences on our environment. For example, they could disrupt ecosystems. They could outcompete the natural organisms that exist in the environment, or they could transfer their engineered genes into the natural, other natural bacteria or microorganisms that will then acquire properties that we didn't intend them to have. There's the issue of biohacking with do-it-yourself DIY biolabs that have been popping up throughout the world, and what is called citizen science, which means non-scientists that are performing scientific experiments. This raises some concern for potential misuse. Mostly, these people that are engaging in this type of science activities have good intentions, and they're using this for good purposes. But there's always the risk that someone in that group could use their skills or equipment for malicious purposes, or simply not really understand the science and the risks and the consequences of what they're doing because they don't have a scientific background necessarily. There's the very important ethical concerns that arise with all of these developments in biotechnology and synthetic biology, which is the ability to manipulate life at the most fundamental levels raises profound ethical questions. And these questions will vary across the globe depending on different groups, religious beliefs, ethnicities, geographical locations, cultural environment, as well as many other factors. But for instance, where should the line be drawn between therapy and enhancement in future endeavors? Should we consider or allow people to enhance their musculature so that they could be superhumanly strong versus trying to cure a musculoskeletal condition that will render someone unable to walk or function normally, or live a life that doesn't require the use of a wheelchair, for example. Where do we draw the line between these two, right? Where do we draw the line between developing a technology that could potentially cure blindness versus giving superhuman vision to some people? What are the implications for creating entirely synthetic organisms that don't exist before? Some of these things we should be thinking about ahead of time. And it doesn't mean that we shouldn't do it. I'm just raising the fact that we should be thinking about it ahead of time so that we don't end up doing something and then in hindsight thinking, oh, we should have never done that. But now it's done and it's not reversible, right? Once you create something and put it out in the world, you will never be able to take it back. And then there's equity concerns. This is a very sensitive issue across the globe, and I'm sure for you students specifically, maybe is particularly significant, which is the risks and the benefits of these technology and technological developments, especially in economical sense, in terms of healthcare sense, and just development of a country. It's been very sensitive because historically, it's been more developed countries that have had access to these technologies and almost to the exclusivity of the technology for many decades. And also people that are able to afford these most costly treatments or this most costly technology, or the people that can travel to a distant place to be able to get a medical treatment that maybe is not available in their town or in their province or their country, right? And then this arises the concerns of, will we be exacerbating the already existing inequities that are in the world, in health, in well-being, in the way that we treat our environment? And so how does that play into biotechnology and synthetic biology? But also the flip coin of that is that, in my opinion, these developments in biotechnology, synthetic biology, and some of the other things I will talk about in a few slides, actually even the playing field. It will make access to some of this technology more easily accessible to countries or institutions or scientists that maybe historically could not have access to that. One example of that is artificial intelligence, AI, right? So there's two terms, biosafety and biosecurity. So I just wanted to talk about what their differences are, but they overlap because they also go hand in hand. And I also want to clarify that when we talk about biosecurity, there's really two types of biosecurity that the world thinks about. One, biosecurity is sort of biosecurity at your farm, biosecurity at your borders, at the airport, where maybe the country or the region or the farmers are trying to control particularly diseases that might harm cattle, sheep, the environment from penetrating your borders, right? Or if you have an outbreak in a farm, you're trying to prevent that from spreading to another farm. But the other biosecurity, which is really what I'm talking about here, is biosecurity in the context of a research laboratory in terms of containment. So biosafety is really concerned with the safety of the scientist or the person conducting the research. So biosafety is concerned to making sure that the person doesn't get exposed to something or infected with an infectious disease that may develop then a condition, right, or a disease. Biosafety manages accidental risks. It attempts to minimize risk from engineered biomaterials, for example, and it's guided by precaution and risk assessment. Biosecurity, on the other hand, especially in a laboratory setting, is concerned with the safeguarding of what's called valuable biological materials. Valuable biological materials could be anything that you generate in the process of doing your research. It could be a plasmid, it could be a genetic construct, it could be a new cell line, it could be a new bacterial strain, or a new type of mouse, or a new disease model, right? It could be new antibiotics, antibodies or antibiotics that you developed. It could be a new biomedicine. It also includes your data that's stored in your computer, your sequencing data, your analysis data. So biosecurity addresses the intentional misuse or theft of these materials. It covers security measures, sometimes physical security measures, sometimes cybersecurity measures, so that you can control the access to your facility or your agents. It's driven by a need to prevent intentional harm, but they overlap in that they both require a risk assessment, training and education, a regulatory framework by which you have guidance, and collaboration and information sharing between the two, between different labs, between institutions across countries and global regions. So when you do a biosafety risk assessment, you review the biological agents and properties, the infectious dose, if it's zoonotic or not, the mortality, the morbidity. The mortality is what's the percentage or the likelihood that a person will die, or what percentage of the population will die from this infection. The morbidity is how sick might you get from the infection and what might be the long-term side effects. Excuse me. Then you looked at the planned activity, which is what is the experiment that you're going to do? The risk is different if you're going to test a clinical sample for diagnosis. For example, I know in India, Mycobacterium tuberculosis is a big issue. It's a problem, especially multi-drug resistant TB. So it's very different if you have a TB sample that you collected from a patient that you want to test whether they have TB or not, versus you're going to actively grow the TB bacterium and then conduct some experiments to try to come up with an alternative treatment, right? The risk is much higher when you're culturing large volume of this bacteria. And the containment would be different because this bacteria is infectious through the aerosol route. So you need to control the air, for example. And then based on this, you would determine what's the appropriate biosafety level measures, the containment. Do you need to work in a biosafety cabinet? Do you need negative air pressure? What kind of PPE would you wear, right? In a biosecurity risk assessment, you would look at what are the fundamental agent properties and could it be used for malicious intent? Could somebody try to steal your TB sample and aerosolize it and spread it across some population, right? And if they did that, what would the consequences be? But also, what are the risks that the sample could accidentally escape the lab, let's say through your biohazard waste or through your liquid waste, right? And again, you look at the planned activity, right? What are you doing? What are you planning to do with this? If you're just having a TB sample that you're testing for TB, even if it's positive, the amount of bacteria in the sample would be very small compared to if you're culturing very large volumes of TB for research purposes, right? Someone with nefarious intent could use a lot of your higher volume concentrated bacterium versus a TB sample that you collected from a patient. So based on that, you would determine the appropriate biosecurity measures and level for your operations, right? Meaning, do you have to have a locked door at your lab or maybe your building? Do you need to have a security badge where people have to swipe? Or maybe you have a security guard that guards the perimeter. Maybe you lock your sample in the freezer with a locked box. Different things that you could use. Together, these two define the lab operating environment in which the scientists operate. Now, I know you're scientists, you're biotechnology students, but I just wanted to frame this around you because when the safety people come around, it might not make sense to you. So I just wanted to explain why we do these things and why it's important. So when you do a biosecurity risk assessment, you always want to think ahead prior to the exit, prior to an incident happening, right? You need to think ahead about what could happen. But it's not just what could happen because you don't want to exaggerate the risk, right? So you want to look at what could happen, but what is the probability or the likelihood that something actually will happen, right? And what are the consequences of that? In some cases, the probability will be very low, but the consequences will be very high. And then you need to decide, am I willing to accept this risk? And remember, there's risk acceptance and risk tolerance. And that could be at the personal level as an individual. It could be as your lab level. That's, you have your individual labs culture for your particular group. Then you could be your department and your entire institution, right? Some groups are more risk tolerant than others. Some people are very risky and very comfortable with taking risks, and some people are not. But keep in mind that when you're doing research at an institution, it's not about just your individual risk tolerance, but also the risk tolerance of the institution, because they have to guard, for example, the institutional reputation with the community. You want to make sure that you don't accidentally release something infectious or potentially damaging to the community where your institution is located. And then you will lose the trust of the community that you're doing everything that you can to protect them while you're performing your research, right? So you define the probability of having an incident based on all relevant factors. Consider the properties of your assets, the potential for misuse. And then you come up here with your plan, and then you implement mitigation measures for that, which means you try to reduce your risk. During an incident, you want to try to identify the root causes and if there were any gaps that you missed during your biosecurity risk assessment. Now, in case of a biosecurity risk assessment as opposed to a biosafety incident, usually in a biosafety incident, you know the incident occurred right away because somebody maybe poked themselves with a syringe that was loaded with something infectious, or maybe your mouse escaped or something like that. But in case of a biosecurity incident, if some material was taken without your knowledge, unless you're keeping very strict inventory, for example, it might take weeks, months, or even years to notice that something went missing. So you may not really know it straight in the moment, but you want to identify your root cause and your gaps as soon as you know that the incident occurred. And then post-incident, you want to look at the consequences of the factors that affected the incident, or anything that incremented this or increased the severity of the incident. You want to consider what the asset loss and what were the impact of losing that asset. There's economic impact, there's psychological impact, political impact, financial, and like I said, reputational. And then you want to regain control of the material that you lose as soon as possible, if you can. It's not always possible though. So now let's talk a little bit about more futuristic things. I want to talk to you about artificial intelligence, humanoid robots, and lab automation. So artificial intelligence is the simulation of human intelligence by machines, computers, that are programmed to think like and mimic human actions. AI technology can hold immense potential to revolutionize many industries, and it already has been in the past 2 years. That can have impact on everyday life. It can offer many solutions for mundane tasks, but also for complex problem-solving scenarios in research. For example, there's an AI now called AlphaFold that can solve the 3D structure of a protein just with an amino acid sequence, something that a few years ago was deemed nearly impossible. In synthetic biology, AI is a very important tool that can propel the field forward very fast, as well as biotechnology. So I developed in a paper that I published last year a biosecurity risk assessment for when you're using AI for your synthetic biology or biotechnology experiments. Let me see. Does anybody have any questions so far? Okay. Let's continue. Can you see my screen still?
Yes, we can see, Leyma.
Okay, perfect. So within this biosecurity risk assessment that I developed, there's 7 easy steps. I won't really go into depth here today because there's other things I want to discuss, but I give you the reference and you can always go to my paper and look it up and ask me questions. I developed a risk assessment matrix. For this, I modified it from the WHO Biosecurity Risk Booklet. You can find it in my paper as well. In this paper, I did a risk assessment for ChatGPT. So what I did want to do is ask a quick question of, what do you think the risk level for using ChatGPT for at that time in, let's say, helping you develop a new, more resistant Shigella strain would be? Anyone?
Uh, Hello, ma'am. Can you hear me?
Yes.
Yeah. Ma'am, I think the risk, myself, Rishikesh Gupta. Ma'am, I
Yes.
think the risk might be nowadays the people are getting reliable on the chat GPT, due to which the work efficiency of the people and the developing strategy that the humans are not developing right now. Like if I want to make something, if I want to do something, so we are just reliable on that chat GPT and we are reducing our efficiency. So the human efficiency is reducing, plus as well as the new thinking which human can, while we are working, during our work we think about multiple things, we get to develop multiple things, but if we
Yes.
are not working, so we cannot think about the new ideas also, so these things are all lacking just because of that.
And do you agree with Ive's opinion?
Students can unmute and respond
Yeah,
to
yeah.
Leyma. So we want to have an interactive session. So don't shy to unmute yourself.
Yeah,
And
don't be shy. Or,
yeah,
feel free to type in the chat if you are not able to speak.
yeah.
Yes, we agree.
Mhm.
You agree? Well, let me tell you, many scientists are using artificial intelligence in their research every day. It's not necessarily going to impede your thinking. It can help you speed up your research process. There is a difference, though, between if you are an expert at a topic versus a novice. So a person who is a novice and doesn't understand something deeply already on their own, they may be using AI such as ChatGPT as a clutch and deferring their thinking, their critical thinking, to the AI. But a person that's already an expert, for example, can use ChatGPT to speed up your process, right? So, for example, you can use ChatGPT to help you craft a reply to a message that could be technical, something that could have taken you an hour to 2 hours to maybe 5 minutes, right? And because you're the expert, you can revise the material provided and say, yes, this is right. No, this is not right, right? You already know the material, you know the content, is just speeding up the time that it would have taken you to look up the book, look up the information, put it together, right? Similarly, in the lab, AI is helping speed up analysis. So for example, before I worked at my current company, I worked at the California Institute of Technology, Caltech, in Pasadena, California. There, the students are developing their own AIs for tools to accelerate their research. Let me give you an example. A student developed an AI that can help them count cells in the microscope. So let's say if you're doing some microscopy experiment where you stain your cell with something to look at your specific protein or an organelle, right? And then you would have to count hundreds of cells, if not thousands of cells, so that you can gain statistical significance for your results, right? Well, this student basically trained the AI to do that for him, and then he didn't have to do it, right? And that's a very monotone, mundane sort of task. You don't really need to be counting hundreds of thousands of cells for hours and hours. You already know what you're looking for, right? So this is a way that it can speed up your process. There's also different types of AIs that can help you model potential inhibitors, chemical inhibitors for a particular protein pocket. And then it can go through hundreds of thousands of potential candidates and yield back, let's say, 10, 20 candidates. So instead of you running libraries, chemical libraries with thousands of potential chemical candidates, that could take you months to do physically, and also will exhaust a lot of reagents. It can give you a smaller pool of potential candidates that then you can focus your time on that will likely be more successful. Right? Let me go back to my presentation now. But just something to keep in mind. So in my paper, actually, that I published, I determined the risk level for ChatGPT to be very low. Meaning ChatGPT is not, it's unlikely that it will be giving you ideas for creating a new bioweapon and feeding you the protocol for how to grow and purify this new bioweapon that you will create. In fact, when you try to do that, ChatGPT will not do it. So you can use it to speed up certain tasks, to learn information. It's not as scary as it sounds. The other topic I wanted to discuss is the topic of using robotics in the lab to optimize and speed up the process of science. So this is a figure from a paper I published this year on the idea of using humanoid robots in high containment laboratories, BSL-3, BSL-4. And this is just a graph that maps the evolution of robotics in the lab, starting with the first generation of robots that did single task, right? So you have automatic pipetters, or we had sometimes robots that poured your Petri dish plates by the hundreds. But those robots were only able to do one task. Then we had second generation robots where you had maybe different robotic arms that could do multiple things. So you might have already seen them. You may already have them at your university, where you have a certain robotic setup where it goes through, it can do a mini prep for you, extracts your DNA, and then maybe you take the DNA and you take it to another robotic section, and you might do some PCR, right, with high volume. These types of setups were used a lot during the pandemic when they were processing extremely high volumes of clinical samples for diagnostic testing. The third generation is what I called cobots, or co-working robots. These are the little robots that you might see in a fancy hospital. They move around. They might carry samples. In this picture, this is from a company that the robot carries samples from one place to another. It may carry charts, or it may help do some other mundane little tasks. There are little cobots that can help disinfect a patient room, for example. And the fourth generation, which we are just starting now, is the embodied AI agent, which is a humanoid robot, a robot that has human shape or similar to a human shape, that has AI embedded in it. So, for example, ChatGPT is not embodied AI. The ChatGPT that we use lives in the computer. There's this thought that true learning and true intelligence involves experience. Yes, so here I have a picture of the first generation, single, this is just a single air liquid handler, right? It'll just pipette a different volume. Here is a very cool picture from a company, but it's basically different robotic arms that each performs an individual function. but together in a row produce a series of functions that can carry out a protocol, for example. And then we have the little cobots that move around. And in here, here's an image of a Tesla humanoid robot that is putting little bottles in this case. So these humanoid robots, Tesla is not the only company, but they can be integrated with artificial intelligence, and that can propel artificial intelligence models to learn even more, right? Because they will be out in the real world. They can experience, for example, tactile function, which is something the robots still struggle with. So this is not fully completely ready to go yet. It's a little bit futuristic, but this area is developing very fast, right? They can walk around, they can pick up boxes, they can pipette, they can do different simple tasks. Also, they can have cameras in them, they can have sensors, so they can collect data as they're moving around, data around the lab. You can imagine, for example, in a lab with significant pathogens that are serious, if there were a lab spill, instead of using a human or requiring a scientist to clean up the spill, you could train your robot to clean up the spill for you. And thus you would be reducing the risk of the human to expose themselves to the whatever pathogen was spilled. These robots could collaborate with humans. So I defined 4 basic types of human AI agents that are embodied or robots, and how they could interact. And actually, I didn't really invent this for myself. I just adapted it to biotechnology, but this has been discussed before in other fields, like using robots for manufacturing, for example, car manufacturing, using robots in factories, things like that. So you can have robot-human coexistence, where the human and the robot work together in the same space, but they're not collaborating, they're not talking to each other, they're not performing anything together. You can do synchronization, where the robot and the human are working on the same thing, but in separate ways, and they don't really overlap. Or you can have robot-human cooperation, where maybe the robot can hold something for you while you manipulate a sample, something like that. Or the robot can be conducting some readings. And then you can do collaboration. The simplest example of collaboration is you can have a robot help you carry a very heavy box. That would require you and the robot working together to carry out this task. To me, this has a lot of potential to revolutionize research in the lab, because you could train these robots to do a lot of mundane tasks that can be done quickly. They're repetitive. The robots can be trained to do it very accurately. They usually do not deviate from protocols. They can work overnight. There's many advantages to using these robots. The main obstacle right now is the cost, right? The return on investment is not something that's justifiable just yet for this idea, but it's only a matter of time. And there are companies that are already using this type of technology in their research, or have done proof of principle and proof of concept experiments. For example, Google DeepMind, which is a branch of Google company, they have a laboratory branch. They published a paper where they conducted a series of chemical synthesis experiments. For 17 days, they used robots in a self-driving lab, which is completely automated, no human intervention laboratory. For 17 days, they conducted over 300 experiments, and were able to synthesize over 40 new chemical compounds that did not exist before. And the robots were able to characterize the chemical properties of these compounds. The robots failed to synthesize some of the compounds because they needed optimization in the protocol, right? But the accuracy was pretty good for a first experiment. You can also envision this type of setup being used in manufacturing of pharmaceuticals or biopharmaceuticals, where you're not really conducting research and development, changing the conditions of your experiments, but you're really pretty much doing the same procedure over and over again in the same manner, and it needs to be done exactly as you say it needs to be done. You might be using large bioreactors where it might be harder or more dangerous for a human to be in there versus a robot, right? Where a robotic strength may supersede the human strength. And also you may be removing the human from some physical risk. Self-driving labs are coming. They're being slowly piloted in academic institutions, but we need to do risk assessments on these labs. Yes? Okay, so in order to integrate a self-driving lab, it will enhance your research efficiency, but it also pose new security challenges. Of course, there's always a challenge that you might get hacked, right? Or some nefarious actor might try to come in and manipulate your experiments for their own gain. They might try to steal your data. So you want to look at systematic risk assessment where you identify the threats, you look at the vulnerabilities, you do a risk evaluation, and then you will have to develop mitigation measures and continually monitor that. I discussed this idea more in detail in a book chapter that hasn't been published yet, but it's in press. But if you have more questions, feel free to reach out to me and I'll be happy to discuss with you. And then we would need a security framework for this. Yes?
Sorry to interrupt you, ma'am. As you
Yes.
mentioned in the topic, you would be reducing the human risk to explode themselves from the laboratory,
Yes. Yes.
then collaborating with humans. Then I
Yes.
have a question. Do you see robots eventually replacing humans' presence in the high-risk labs, or even will they always act as a support tool for scientists?
I think that robots will be there to support the scientists. I don't think that we will ever have high containment research fully devoid of humans, at least not in the next 50 years, in my personal opinion. But I think these robots can greatly enhance the work that we already do, especially when it comes to monotone things or monitoring things like temperature, fluctuations, humidity. Taking not only that, right, because we already have sensors that take that information, but if you combine that with the AI integration, these robots can really analyze this data for you very quickly, right? So they can find anomalies in the system. They can say, hey, the negative air pressure system in our BSL-3 is fluctuating, and I notice it's fluctuating at this time of the day or on this day of the week, or when we're doing these other experiments. And it might be able to find correlations with other things that maybe a human being wouldn't be able to notice, just simply because of the capacity to hold the data, right? If you have a human analyzing some, let's say, temperature sensor information, and if they're doing it by hand, it could take them hours to do that. And it will detract from actual research, right? You would need to have a person doing that where they could be doing something at the bench. The humans will also need to do the creative work because the AI and the robots integrated with AI, they really don't have the creative capacity to think in the way that we do. They are more, the way I view them right now is, at best, they are like a very smart student that memorized the entire textbook. And if you ask them anything about the textbook, they can tell it to you immediately. They can recite things. But if you ask to synthesize new information or put together unrelated topics to create a new idea, they really struggle with that, right? And we humans can do that, of course, with training and with acquiring certain levels of expertise, not a novice student, but eventually with more training, you will get there. I know that this also sounds scary because people say, oh my God, they're going to take our jobs and then what are we going to do? Or these robots are going to take over and do who knows what. Maybe they're going to be running their own bioweapons program. And that's why this is important. And this is important, right? Because we humans are the ones designing them. So we get to set the parameters of what they do and to what extent they go. But we shouldn't leave this in the hands of non-experts coders, for example. As scientists, we should participate. We need to be well-informed and educated enough to be able to speak the language, and then get together as a group of scientists locally and globally. And I think you as students are the perfect time to do it, right? Because you're still forming yourselves as scientists. So we have to think about scientific research, regulatory compliance, but we regulate ourselves, right? We get to decide what we think is right and what we think is wrong, what should be acceptable and what shouldn't be acceptable. And then we get to form frameworks around that, right? So then you get to do security by design, so that you design the system so that it's inherently safe. You design the system so that it complies with our regulatory frameworks that we have. You design a system that will behave ethically and will have ethical research practices. You also don't want an AI robot that will run away and create new data that didn't really exist and just basically manipulate the researchers, right, by fabricating data that from experiments that were not really run. But we get to create that framework, not them. We are designing them. So we have to be involved, and we hold responsibility for them. If we don't have them, if we don't want them to run over, take over, and do extremely unethical things, we have to set the parameter around it. And I'm calling attention to this because I think that at a world stage and regionally in almost every country, the regulatory frameworks are running really slow and really behind, and the technology is moving really fast. So by the time some government regulator decides, oh, we should do something about this, it will have already been done, right? And that's what I want to try to mitigate. So usually, yes?
But considering the rapid advantage in the AI robots, do you think it will be carry out complex experiment and decision-making processor without human overrides?
They shouldn't do it without human override. But could they carry out complex experimental processes? Yes. I think with training, they can.
Thank you, ma'am.
Yeah. So normally,
Ma'am.
Yes.
Sorry for the interruption, ma'am.
No
Ma'am, my
problem.
name is Shubham Sharma. Ma'am,
Yeah.
as we hear the word robot, we always think about the stuff that robots are used in heavy duty stuffs in industry. So my
Yes.
question is that the robot can be that much precise that they can be used in techniques like gene editing and gene therapy, because this all
Yes.
are high laboratory works. So can robot be used in these techniques?
Yes. They can with your help. But let me change your context a little bit. There are robots that are conducting microsurgeries for surgeons currently, where before you used to cut up a patient, open up and make a big incision and move organs around. And now these robotic hands can go in and perform these surgeries at the direction of the surgeon, right? And in some cases, it's been reported the surgeon was not even in the same country as the operating room with the patient. So this has already been done. Some of these surgeries have occurred for in utero operation of a human fetus that hasn't been delivered yet, where they did heart surgery to fix a heart defect for a yet to be born baby, right? So yes, these robots could potentially do highly complex experiments, probably even better than a human would, because they wouldn't have pipetting errors, for example. They wouldn't forget the order of the reagents, or whether they forgot a reagent, or whether the temperature was off, or, you know, I ran out of this material, but I'll do it anyway, right? The robots wouldn't do that. You hold the parameters, so they would have a very strict SOP that they would follow, and that's it. That's all they do. A risk that concerns me a lot, actually, is that countries or terrorist organizations could try to retake up, let's say, bioweapons programs. That is completely illegal to do, but they could do it with completely automated robots, because the footprint for such a laboratory would be much smaller. And if you have fully automated laboratory for such nefarious intents, remember, the robots can work 24/7. The robots don't need the lights on. You could have the lights off. But also, the robots do not talk to other countries' spies. They don't have moral disappointments. You know, there's no human factor that could be a sort of weakness in a country's bioweapons program, where scientists could say, this is highly unethical, I'm going to do something about it, right? So this is also a concern. But you can also imagine using these robots in countries that maybe was way beyond their reach in the past, once the cost is lowered. So normally we conclude with conclusions, but I thought today I would leave you with thought-provoking questions. So these are questions that I don't have the answer to. These are questions that I think about, and I would like you to think about too, individually or as a group. You don't necessarily have to do anything about them. I'm not leaving you any homework other than, you know, think about them. We ran out of time today. We're running a little late. So, but I want you to think about, what are the potential benefits and risks of using these embodied AI agents to replace humans in high containment labs, which is the question that we just had a few moments ago, right? What are some dangerous tasks that these robots could do instead of the humans, so that we can lower the human risk? And what else could they do in the lab that we have yet to imagine? And then how might this integration of robotics and AI into scientific research transform the speed of the discovery and the types of security threats that then we must guard against, right? And then, if self-driving labs can conduct hundreds of experiments with minimal human oversight, what responsibilities and what ethical boundaries should we maintain, and which ethical boundaries should we set, basically, for these machines? And with that, I will leave it up to more discussion or questions if you have any.
Alicia, you can go ahead.
Thank you very much, Dr. Leyma, for that highly insightful and thought-provoking talk. You have illuminated the critical intersection of innovation and security in today's biotech era, an area where awareness is just as important as advancement. We now move into our interactive question and answer session. We will try to take as many questions as time allows. To begin, Dr. Leyma, I would like to start by asking, in your experience, how prepared do you think the world currently is to handle potential biosecurity threats that might arise from synthetic biology? We hear a lot about the promise of this field, but the risks are just as real. I would love to hear your take on where we stand globally.
I think we are underprepared. We could be better prepared, and we could be spending more time preparing ourselves. I don't know that we will ever be fully prepared for what is already here and what's coming, because it's just developing so fast, right? But we are going at turtle speed right now. In the rabbit and turtle race, I know the turtle wins at the end, but we are going at the turtle speed, and we need to speed up a little bit. I think where we are going the most slow is that most countries and scientists in general, we tend to have sort of the old-fashioned mindsets. You know, it's very common to find in a university a professor that's been there for
Dr. Leyma, you are mute.
Can you hear me?
Yes.
Yeah, so you may find scientists that are still conducting experiments the old-fashioned way. Meanwhile, there's other groups that are going very fast and advancing very quickly. But if we collectively don't acknowledge that this is happening and think about it before, then we're not going to be prepared, right? Things will happen, and then we will say, wait a minute, who did that? Who allowed that thing to happen? Why didn't we have something in place? But it is our responsibility to think about that and have something in place. I know it's a challenge globally to try to get governments to move fast or try to get legislation approved. I know it can take years, but I always say to scientists when I speak to you, it doesn't have to be the government, right? It could be you, or it could be a scientific organization within your country, or even within your university. You can begin by proposing a framework, starting to set some guidelines or some best practices. Even if you're not legally mandated to do this, you can have some sort of guardrails that you collectively decide that work for you, and then you all agree that this is what we will do. Even if the government is not mandating that we do this, we do it because it's the right thing to do. It's the moral thing to do, right? And the other thing is that this needs to be adapted to each country and each region. I do notice that people sometimes say, well, the US or the EU hasn't published anything. And I say, don't wait for the US or Canada or the European Union to come up with a document. You lead, you know, you can lead too, and set the gold standard. Take the lead. Don't wait for the traditional way of doing things, where maybe the World Health Organization will set a statement. It will take the World Health Organization 5 years to say something about AI. And in the meantime, AI might look completely different than it looks right now, right? So those are some of the challenges, right? There are other challenges that are beyond us as individual scientists or individual students at a university, that will require more diplomatic work or more policy level work at a government level. And to that, I will say, I encourage you all to try to pursue diverse careers in science. You don't just have to be a researcher at the bench. You could have a science policy career. You could go and become a diplomat. And then you will be an educated diplomat. You will understand the science, and you will be doing your country and the world a benefit, because oftentimes our policymakers don't understand science very well, and we end up with policy that doesn't work very well, or that it's even sometimes obstructive to the research progress, right? So we always want to mitigate the risk, but without obstructing the progress. We need to maintain a balance between the two.
Thank you so much. That was a really thoughtful perspective. It's eye-opening to hear that from someone with real on-ground and policy-level experience. Now, shifting to your hands-on work, could you share some examples of biosafety risks you have encountered while working in high-containment labs, and how those situations were managed?
Yes, I worked in a laboratory that worked with tuberculosis, mycobacterial tuberculosis, Francisella tularensis, or called tularemia, Yersinia pestis, which is the plague from the Middle Ages. In the United States, these organisms are considered select agents, which means they're highly controlled because the government considers that they could be used for terrorism purposes or weaponized. So we have laboratories that work all day, every day with these organisms. They sometimes infect mice with them to study just the biology of the infectious process. Sometimes they're trying to develop new treatments, antibiotic treatments, or bacteria for viruses. I worked in the same lab, also worked with Ebola virus and Zika and dengue, for example. So We never had an exposure where somebody actually became ill with any of these diseases. There was a lot of monitoring, offering vaccines when they're available. So, but for example, for TB, that was something that the US takes a lot of care because it's not such a widespread problem in the United States. So we're always monitoring the scientists that are working with a bacterium to make sure that they don't seroconvert, which means that they might have been exposed because the disease doesn't develop sometimes for 10 years, right? We also did work with rabies diagnostics, and rabies can be quite dangerous virus because there is no real treatment. There's prophylactic, but if you develop the disease, the mortality rate is quite high. But there's always issues of PPE failing or human factors where the scientists maybe didn't wear the PPE or didn't wear it properly. And sometimes there's accidents, like you poke yourself with a needle or the animal bites your finger or things like that, right? So the main pattern in these issues that arose in the lab is usually they happen at the end of the day when you've been working for a long time, or at the end of the week when it's tired, you've already had a long week, and maybe you're under pressure, your professor is asking you for a result, or you want to publish a poster, you're going to a conference. I had a scientist that had an incident at midnight on a Saturday, and she was supposed to be on a plane at 5 in the morning the next day for a conference. And she was still collecting data for her poster. And so I just say, be mindful of those things yourself. You know, if you're tired, if you're not feeling well, if something personal happens, you know, maybe you're sad, maybe somebody in your family died, maybe somebody got diagnosed with a serious illness. Maybe you had a problem with your boyfriend or girlfriend or your friend. You know, it's okay to say, I'm having an off day today. I shouldn't be in the lab, right? Maybe I'll go home and analyze my data on the computer, or I will sit here and analyze my data, but I won't really touch the pathogen. So I encourage you all to be self-aware of, are you tired? Are you emotionally well that day? Do you have the mental energy to be alert enough? Because it's not only keeping yourself safe, but keeping your colleagues safe as well. You know, sometimes an accident can result in someone else also being exposed to something that you didn't intend.
Thank you for sharing that. As we conclude today's session, I would like to extend my heartfelt gratitude to Dr. Leyma for sharing her expertise and perspectives with us. Your clarity and depth of experience have provided our audience with valuable insights into how science can evolve safely and responsibly. To all participants, thank you for joining us and contributing to this critical discussion. We hope this session has sparked further curiosity and commitment towards safe innovation in life sciences. And now I would like to hand the session back to our MC. Mr. Nityansh, over to you.
Thank you, Alicia, for moderating the session. As we draw this exciting session to a close, I now invite Ms. Pariti Sharma to deliver a vote of thanks on the behalf of our entire organizing team. Pariti, over to you.
Good evening, everyone. It is indeed a privilege to deliver the vote of thanks on the behalf of our organizing team and CSRD People's University as we conclude today's session of our BioConnect webinar series. First and foremost, I extend a heartfelt gratitude to our esteemed guest speaker, Dr. Leyma P. Haro, for taking time out of our schedule to share such valuable insight with us on biotechnology and synthetic biology in biosecurity era, opportunities, risks, and safeguards. Dr. Leyma, your expertise and engaging presentation have greatly enriched our understanding and added immense value to this webinar. We express our deepest thanks to our Honorable Vice Chancellor of People's University, Dr. Harish Rao, for taking time out of his busy schedule to deliver opening remarks today. We are also honored by the presence of our respected dignitaries. Your constant encouragement and recognition of our efforts are what motivates us to keep striving for excellence. Thank you for joining us and lending your gracious presence to today's session. A heartfelt thank you to our guiding light, our mentor, Professor Dr. Subhash Bore. Sir, your support behind the scene, your faith in us, and your invaluable guidance have been the backbone of this entire series. We are truly grateful for your mentorship. To our moderator, Ms. Alicia, thank you for moderating the session. Your ability to keep the flow engaging and focused is commendable. We also owe our sincere thanks to the chairman of our organizing committee, Ms. Vanshika, for your support during the program. Allow me to take a moment to reflect on the significance of our BioConnect webinar series for the students, by the students. In a time where knowledge sharing transcends physical boundaries, our series has served as a vital channel for learning, collaboration, and inspiration. We have had the opportunity to explore new ideas, interact with experts, and grow as a community of learners. To our audience, thank you for your enthusiasm and participation. Your curiosity and thoughtful questions brought life to today's session. Let this be a reminder that learning never stops, and together we can continue to inspire one another. Last but not the least, we want to record our special thanks to the management of Pupils University for the full support to our BioConnect webinar series. With that, I now hand over MC the program to continue with the proceedings. Mr. Nityansh, over to you. Thank you.
Thank you, Paridhi, for expressing our sincere gratitude to all the respected judges, our expert speaker, and every participant, fellow students, and esteemed participant. Before we signing off, we request all the participants to kindly take 2 to 3 minutes to fill out the post-webinar feedback form. The feedback form link is now available in the chat box. Your responses are extremely valuable and will help us to make future events even better. We hope you have completed the feedback form. Please note that your e-certificates will be sent to your registered email ID shortly. We sincerely thank you once again for being the part of this entire session. We're looking forward to welcoming you in our next BioConnect webinar series very soon. Until then, stay curious, stay inspiring, and keep connecting with us. This is Nityash Patel signing off on the behalf of the organizing team at People's University, Bhopal. Have a wonderful evening.